So here is the comprehensive RNA-seq transcriptomics analysis pipeline. For this study, we have followed this paper by Chen et al. 2022, where they have done the transcriptome analysis that revealed differential transcription in tomato following inoculation with Rastonia solanacearum that causes bacterial wilt. The first step for transcriptomics analysis is RNA-seq data retrieval and pre-processing. So for this, I downloaded the raw RNA datasets from NCBI SRA databases. After retrieval, I performed an initial quality check using FastQC to examine base quality, GC distribution, and adapter contamination. Then, Trimomatic was used to remove adapters and low quality regions to improve downstream alignment accuracy. After confirming the quality of cleaned reads, I downloaded the tomato reference genome from Soul Genomics Network and indexed it using BWA, which prepares the genome for efficient read mapping. The next step is read alignment and BAM processing. So, the high quality reads were aligned against tomato reference genome using BWA-MEM. This step identifies the genomic origin of each read. The resulting SAM alignment files were then converted into BAM format for compressed storage and faster processing. Next, BAM files were sorted and indexed using SAM tools, which is essential for transcript assembly and visualization. Finally, the tomato genome annotation file in GFF format was included to guide gene and transcript identification. Next, transcript assembly and quantification was done. So, in this step, string tie was used to reconstruct transcripts and estimate their expression level in each sample. Since multiple samples were analyzed, all transcript assemblies were merged into a unified non-redundant transcriptome. This improves consistency across samples and helps identify both known and novel transcripts. Then, prep.de Python script was used to extract gene count and transcript count matrices, which served as the input for DE-Sec2 based downstream differential expression analysis. And finally, DE-Sec2 was used for normalization and statistical testing of differential gene expression. Based on log to fold change and adjusted p-values, significantly upregulated and downregulated genes were identified. To interpret the data visually, we generated PCF plots to examine sample grouping and reproducibility, volcano plots to summarize significant DEGs, and heat maps to visualize expression trends among the most important genes across all the samples. This PCA plot provides an overview for the global variation in gene expression among all the samples. The samples that cluster closely together indicate high similarity in their transcriptomic profiles, which confirm the good reproducibility among biological replicates. In the analysis, the susceptible and resistant samples formed distinct clusters, suggesting that the infected condition caused major transcriptomic changes. The percentage values on PC1 and PC2 represent the proportion of total variance explained by each principal component. Since PC1 explains the largest variation, the clear separation among these axes indicates the infected condition is the major source of expression differences. The volcano plot summarizes all genes based on their fold change and statistical significance. The x-axis represents log to fold change, where genes on the right are upregulated and those on the left are downregulated in the infected sample compared with the control ones. The y-axis represents the negative log10 adjusted p-values. So genes appearing higher on the plot are more statistically significant. The red and blue points represent significantly upregulated and downregulated genes respectively, while the gray points are non-significant genes. This plot clearly shows the magnitude and significance of differentially expressed genes, allowing us to rapidly identify the most responsive candidate genes. The heat map shows the expression pattern of the most significant differentially expressed genes across all the samples. Each row represents a gene and each column represents a sample. The color gradient indicates relative expression levels where red represents the higher expression and blue represents the lower expression. Samples with similar expression profiles clustered together, which again validates the reproducibility of the replicates. The heat map helps visualize coordinated gene regulation and reveals group of the genes that may participate in the pathogen response mechanism. So overall, the PCA, volcano plot and heat map collectively confirms the pathogen-induced significant transcriptomic alterations leading to the identification of key candidate genes and pathways involved in the response mechanism.